Hi everyone, thanks for coming to check out my video talk at the Beckman Symposium. My name is Michaela TerAvest and I'm an assistant professor of biochemistry and molecular biology at Michigan State University. I'm also a Beckman Young Investigator from the 2018 class, and today I'm going to talk to you about our Beckman funded project on engineering electroautotrophy in bacteria. This project is motivated by our desire to develop a technology called microbial electrosynthesis. This is a technology wherein we would use wind and solar power to provide the energy and reducing power to reduce carbon dioxide to carbon neutral fuels, bioplastics, and chemicals. And we're really excited about this technology because it can simultaneously solve multiple different sustainability challenges. For example, this is a way that we can store power that's generated from intermittent sources like wind and solar and put it into a long-term storage format like diesel or jet fuel. This also represents a way that we can offset other materials that we currently get from petroleum sources like plastics and make them in a more sustainable way. So we think this is a really exciting technology, but what is really critical for us to be able to develop this into an economically viable platform is to have a microbe that can efficiently catalyze this process. And what we need to do this process is what we call an electroautotroph. So you may be more familiar with photoautotrophy, wherein photosynthetic organisms fix carbon dioxide into biomass and other organics using sunlight as the source of energy. For an electroautotroph, rather than using light as the energy source, the organism would use an electrode, in this case a cathode, to provide electrons for the energy source and the electron source to do the carbon dioxide reduction. Now, the question is, does this exist in nature? And I won't go into it deeply today, but the short answer is yes. You can go to environments like, for example, a wastewater treatment plant or a sediment and take a microbial community and find that there are some organisms and some communities of organisms that are capable of an electroautotrophic metabolism. And there are several research groups around the world who are working on developing electrosynthesis using that framework. However, there are some big challenges with doing microbial electrosynthesis with wild microbes. One of them being that the electron transfer rates on a per cell basis are relatively slow, and we don't have the genetic tools for these organisms to be able to make them work better or produce specific organics like a bioplastic molecule. And this can be for multiple reasons. This could be because it requires a whole community to catalyze the electroautotrophic metabolism, and we don't currently have synthetic biology tools to modify communities as a whole. Or it could just be because the individual organism is so different from our current model organisms in synthetic biology that the tools we have, like plasmids and genetic engineering tools, simply don't work for those organisms. So while there's a lot of interest in pushing those organisms forward and developing their genetic systems, my lab wanted to ask a different question, which is, can we build an electroautotroph instead using genetic parts and organisms that we really understand well, rather than starting from something that has this metabolism already? And the platform organism that we've chosen for this project is called Shewanella onidensis. And what's really great about Shewanella is that it does interact with electrodes, although via a heterotrophic metabolism. So Shewanella is able to oxidize organics, as all heterotrophs do, and the electrons from that oxidation are transferred to intracellular carriers like NADH, which then are reoxidized through an electron transport chain that actually terminates at the outer surface of the cell and is able to transfer electrons to solids such as an electrode. And this evolved so that Shewanella can, in anaerobic environments, use minerals like the ones shown in this microscopy image here as their electron acceptor for respiration. Or in other words, the Shewanella cells that are on this mineral are actually using it to breathe. They're using this instead of oxygen. And researchers have been working on Shewanella for over 30 years, and we really have a pretty clear understanding of how this works. So that's why we chose this organism, to see if we can reverse its heterotrophic metabolism to turn this into our electroautotroph. So that's what this would look like. Instead of electrons coming out of Shewanella, we would have electrons going in and generating reduced electron carriers like NADH inside the cell. Now, for that to work, we need a couple additional things. So because we've reversed its respiratory electron transport chain, we now will have the consumption of proton motive force instead of the production of proton motive force. And in our experiments, we don't provide any sort of native electron donor or electron acceptor, so Shewanella doesn't have a way to regenerate proton motive force to keep the electron flow going in reverse. So we've added a protein called proteorhodopsin, which is a light-driven proton pump, to allow them to regenerate that proton motive force. The second thing we've added is an electron sink in the form of this enzyme, butanediol dehydrogenase, and its substrate, acetoin. This is because although our eventual goal is for carbon dioxide to provide the electron sink, it's going to take a lot more work to engineer a carbon fixing pathway into a heterotroph. So for now, we're using this pathway as our proof of concept because we can very easily track the formation of butanediol and get a measure of how effectively our inward electron transfer to Shewanella is working. We then take these modified Shewanella and we can put them in a bioelectrochemical system with a cathode and with green light to power the proteorhodopsin, and we can measure the success of our engineered inward electron transfer pathway in Shewanella. And when we do this, we see that our engineered pathway functions as expected. So here in the plot, you're looking at electron uptake from a cathode over time. And this dotted line shows the current we would expect if there were no cells present in the bioelectrochemical system. And the greater the negative current is, the greater the number of electrons that are passing out of the electrode. So a greater negative number means higher performance of the inward electron transfer pathway. So in the green line, you see that over the course of several days, the engineered Shewanella were able to take up significantly more electrons from the electrode than would be expected for just background current. You can also see these little spikes here, which show times when we turned the LED light off, indicating that the electron uptake was dependent on light, as we would expect based on the design of our pathway. We also checked a version of the strain where proteorhodopsin was simply absent, and that significantly reduced the ability of the strain to take up electrons from the electrode. Similarly, with the butanediol dehydrogenase, if it's now present, the Shewanella have a significantly reduced ability to take up electrons. And if both of those heterologous proteins are absent, there's very little activity for just basically wild-type Shewanella to take up electrons from the electrode, again, as we would expect based on our design. We also tracked the production of butanediol over time to ensure that the electron uptake from the electrode was going where we thought it should. So here you can see that, again, over the course of several days in the green line, our engineered strain with both proteorhodopsin and butanediol was able to accumulate significant butanediol. And when proteorhodopsin was absent, as you see in the purple line, there was a significantly reduced ability both to take up electrons and form butanediol. Importantly, we also repeated this experiment in the absence of the poised electrode to ensure that the butanediol formation was actually dependent on inward electron transfer. And indeed, we saw that there was a significant reduction in 2,3-butanediol when the electrode wasn't present, indicating that the butanediol formation really was being driven by electrons from the electrode. There is some background formation of butanediol, and my lab is continuing to work on improving the experimental conditions to reduce this background, but I won't have time to talk about that today. So the last few plots I showed you indicate that our engineered system, the heterologous proteins that we expressed, are doing what we expect them to do in this system, which raises the next question: which native pathways are we relying on to support this inward electron transfer? Now, it's clear from a lot of past research on Shewanella that this MTR pathway, which I won't get into the details on today, is critical for getting electrons from the electrode surface to the inner membrane of Shewanella. So we didn't dig deeper into that because we already have a very clear understanding of that part. However, what we didn't know was how electrons are getting from quinones in the inner membrane to NADH inside the cell. And the reason that we were so unsure about this is because there are 4 complexes encoded in the Shewanella genome that could potentially carry out this function. And these are NDH, NUO, and NQR1 and NQR2. All of these catalyze electron transfer between NADH, and quinones and quinols in the inner membrane, but they have differing capabilities to couple that electron transfer with the movement of ions across the inner membrane. So NDH doesn't move any ions, NUO moves protons, similar to what you would expect for complex 1 in the mitochondria, and NQR1 and NQR2 pump sodium ions. So we expected that probably NDH would not be involved because the inward movement of ions is likely necessary to drive the reverse electron transfer. So we looked a little bit deeper at these other electron transfer components. And we've made several deletions and deletion combinations to test which of these are important for inward electron transfer. And I won't go into all of the details, but I'll show you that through several different projects in my group, we found that NUO and NQR are generally the most important NADH dehydrogenases in Shewanella under a range of conditions. So here you're looking at aerobic growth, and if either NUO or NQR is present, so here in green and blue, you're looking at the single knockouts, the strain can perform similarly to the wild type. But when both of those have been deleted, which you see in the pink line, the strain can no longer grow in aerobic minimal medium. Similarly, if we look at outward electron transfer, so this is the native process I talked about Shewanella transferring electrons to an anode, again, we see that the wild type in black transfers electrons. Either of the single mutants missing NUO or NQR1 can perform similarly to wild type, but if both of those are gone, the strain really has very little capability to transfer electrons outward to an electrode. So we thought that this double mutant strain lacking NUO and NQR1 probably would be the most promising thing to test for how inward electron transfer is working. So here, again, in the green line, you're seeing our base engineered strain that has all of its native pathways plus proteorhodopsin and butanediol dehydrogenase. And then in pink, you're looking at the double knockout strain that's missing NUO and NQR1. And what you can see is that there was a significantly reduced ability to take up electrons from the electrode when we knocked out those 2 complexes. And there was a complete disruption of the ability of the strain to accumulate butanediol. So these results strongly suggest that both NUO and NQR1 are essential for transferring electrons to NADH, which again aligns with our model of how inward electron transfer should work in Shewanella. Now that we have a platform for inward electron transfer in Shewanella that works consistently and works the way we expect it to, we have several different fronts to move this project forward. One is to remove the requirement for light, because as I said, we want all of the energy to be provided by the electrode rather than from a green LED light. As I also mentioned earlier, we need to engineer in a carbon dioxide fixation pathway because as a heterotroph, Shewanella doesn't naturally have this. And I also have a collaboration with another group to engineer Shewanella to produce interesting products like terpenes. And today I'll mostly focus on removing the light requirement and talk a little bit about carbon fixation. So as I mentioned earlier, we add proteorhodopsin because we need a way to generate proton motive force. But an alternative strategy is to use other native components of the respiratory electron transport chain, like cytochrome oxidases, to catalyze favorable electron transfer reactions, such as the transfer from quinones to oxygen to form water. These complexes can use the energy from those favorable electron transfer reactions to generate proton motive force. So our hypothesis was that if we would add some oxygen to the system and not add light, that we would be able to have electron transfer both to oxygen and to intracellular NADH and be able to catalyze butanediol formation on the electrode without light. And we saw that this was indeed the case. So here you're looking at electron uptake in the absence of light, but in the presence of some oxygen. And what you see is that the strain indeed was able to take up electrons from the electrode. The current was more variable in this case because it's somewhat difficult to control oxygen when it's all around us in the atmosphere. But my lab is currently moving forward to try to enhance control of oxygen in our bioelectrochemical systems. Importantly, the strain was also able to accumulate butanediol over time. And again, there was no other source of electrons in the system than the electrode. So this suggests that indeed electrons flowing into Shewanella can be transferred both to oxygen and to NADH and allow the formation of butanediol driven by the electrode. So moving forward, we're going to be working with controlling oxygen better and optimizing the oxygen concentration to ensure that we can get the maximum amount of transfer to acetoin and the minimum amount of transfer to oxygen. Finally, we are continuing to work toward adding a carbon dioxide fixation pathway to Shewanella. This will require adding a couple of extra proteins, PRKA and Rubisco, to complete the Calvin-Benson cycle for carbon fixation. And I don't have time to get into it today, but my poster is really focused on this aspect of our project. So please check out my poster if you're interested in this part of our project. And finally, I'd like to thank my whole lab. The team that's been working on this project is mostly Nick Teft and Katie Ford, who did all the work that you saw today, but they're really supported in this by everyone else in my lab. And again, I'd like to thank the Arnold and Mabel Beckman Foundation for funding this work. Thanks.